Hello and welcome to Genetics Day 5. Okay, warning before we start this video. This is the most complicated thing we will cover all year. Warning, this is the most complicated thing we will cover all year. So if you are confused, that is understandable. This topic is actually even more complex than it will be presented to you in this video because the complexities of life are numerous. So, again, if you are confused, I'm going to post alongside this video on Google Classroom a copy of this notes sheet with all of the answers on it. So please watch this video as well as you go through your notes so that you can hear my explanations. Is really, in reality, much more complex and simplifying it for our purposes in living environment. So if you are confused, that is okay, and it will not get more complex than this. This is the most complex topic that we are going to cover all year. I will say from now until the end of the genetics unit, and then units 8 and 9 will be actually a little bit more simple to understand. But this is very important because this is how our DNA is actually the instructions for life. This is how those instructions are transcribed, and translated. So today we're going to get to how transcription leads into translation. And again, warning, this is the most complicated things we'll get all year. So let's go ahead and get started. Today's aim: how does DNA and RNA assist in protein synthesis? So how do these molecules actually wind up creating proteins? Before we do that, let's go over these two do now questions. So if 15% of a DNA sample is made up of thymine, T, what percentage of the sample is made up of cytosine? So I keep trying to write on this, but it's not letting me. So forgive me. So I'm going to say some calculations out loud. Let's see if I can annotate them on the screen. Hold on. Wow, no, I cannot. Okay, so if 15% is thymine, there's only 4 bases, right? So we know A matches with T, all teachers go crazy. So another 15% of the DNA sample is also adenine, right? So then that takes away 30% of the DNA sample. We're left with another 70%, but we're not done yet. Out of that 70%, half of it is cytosine and half of it is guanine. So that means the answer is B, 35%. 35% of the sample is cytosine. Number 2, chromosomes can be described as large molecules that have only one function, which, no, that's really DNA is the large molecule, and it is the instructions for life, but that's kind of more than one function, but it's also only one function. I basically think that that answer is referring to DNA, but that's not what chromosomes are. Chromosomes are coiled strands, bundles of DNA. So chromosomes can be described as D, coiled strands of genetic material. So the answer to number 1 is B, 35%, and the answer to number 2 is D, coiled strands of genetic material. Let's go on. All right, so far, the genetic information stored as base sequence has moved from the nucleus to the cytoplasm by using RNA. And remember, we talked about that in day 4. The DNA is transcribed into a strand of mRNA, which is a single-stranded molecule that can safely leave the nucleus and make its way to the ribosome. But now another problem remains: how to use the nucleotide base sequence in RNA to build a protein with the correct sequence of amino acids. This problem involves a change of language, translation, from the base sequence language of RNA into the amino acid language of proteins. This process is called translation, and it occurs at the ribosomes. So we're probably already familiar with translation, whether you speak multiple languages at home and you're translating between them, and like English and another language with your parents, or you just use Google Translate for your Spanish or French homework, we know what translation is. And that is basically what this process is doing. It is translating the mRNA code into the amino acid sequence that will build proteins. And this happens at the ribosomes, which are actually reading this translation? So, well, reading the mRNA and turning it into the amino acid sequence with the help of tRNA, but we're getting there. We're getting there. So, built into every living cell in the world is a genetic code. It is called the triplet code. Each different combination of 3 bases makes up a word called a codon. So, a triplet of mRNA bases is a codon. So, 3 mRNA bases, AUG is a codon. Each codon represents a specific amino acid. Each 20 amino acids has at least 1 codon, and most have more than 1. And this is mutation insurance, but we'll get to that. The genetic code is universal. In other words, all organisms on Earth use the same genetic code. For example, the codon GCA stands for the amino acid alanine in all life forms, from bacteria to trees to humans. This similarity among living things is good evidence that all organisms evolved from a common ancestral life form in Earth's distant past. Wow, that's pretty cool. So, what is translation in terms of protein synthesis? So, translation. So go ahead, pause this video, and using that passage above, annotate it and answer those 3 questions. Did you pause the video? Okay, now let's go over the answers. Hopefully you paused the video. This is the document that I'm going to share with you guys as a PDF so you can go back and reference it. But what is translation in terms of protein synthesis? Translation is the process of using mRNA codons to string together amino acids to create proteins. It is changing the language or translating mRNA into the amino acid language of proteins. Remember, amino acids are the building blocks of proteins. What is a codon? A codon is a combination of 3 bases, basically a word in genetic code that codes for a specific amino acid. And we will use the universal codon chart, which is kind of like a complicated looking chart, but anytime that you actually have to use it, for example, on the regions, you will be given the universal codon chart. And we'll go over how to read it in class when we do our classwork for this day. So we will use the universal codon chart to see which codons code for which amino acids. How is the genetic code universal? All living organisms on Earth use the same mRNA code, which is evidence that all organisms on Earth emerged from one common ancestor in our evolutionary past. So now let's go over protein synthesis notes. So how does mRNA code for proteins? mRNA leaves the nucleus and goes into the ribosomes, which are located freely floating in the cytoplasm or on the surface of the rough endoplasmic reticulum. These proteins are built from instructions on the mRNA. And the ribosomes actually are reading this strand of mRNA. And they will then use their ability to read this mRNA with the assistance of tRNA, which we're going to get to code this amino acid sequence based on that strand of mRNA. And that process that we're about to get to is the translation. So how does this happen? How does mRNA code for proteins? Well, first we start with the DNA in the nucleus. We have the strand of DNA. The DNA is unzipped in the nucleus, and one side of that double helix is used as a template strand to produce a strand of mRNA, which can then safely leave the nucleus. And usually one part or one segment of DNA is coded for at a time. Those segments of DNA are genes. So one gene is taken as the template and turned into a strand of mRNA that represents the DNA that codes for that gene. Stay with me. The ribosome then reads that mRNA, right? The ribosome is the reader. And then a protein is constructed by using specific amino acids that are coded for based upon those codons, those 3 base sequences that are used to code for specific amino acids. And then those amino acids are strung together to form a protein. And how can we code for 20 amino acids with only 4 DNA bases, right? The math doesn't seem to add up there. Well, mRNA codes for proteins in triplets. And again, this is where that universal codon chart comes in. So the ribosome reads the mRNA, and then based upon what it reads, using the transfer RNA to assist, the amino acids are put together in sequence and then linked together to form proteins. So we can see there, AUG codes for methionine, CGU, etc., codes for the different amino acids shown below. Those are codons that code for specific amino acids. And a codon is, again, a block of 3 mRNA bases. But the process is actually a little bit more complex than that even. So this is the universal codon chart. It looks confusing, but we will talk about how to read it in class. But there's a couple of things that you need to know about it. So the mRNA code is the same in all life, which is like literally amazing. This is the strongest support for the common origin of all life. The code has duplicates, which means several codons code for each amino acid. This is mutation insurance. This helps prevent mutations from actually changing the protein structure. So let's say at some point in the process of transcription or translation, or even in DNA replication, one base is switched for another base. Well, if that happens, multiple codons code for the same amino acid, so that ultimately the protein construction, which is actually what determines our traits or characteristics, are not altered. If it is a small mutation, and we're going to talk a lot more about mutations on day 6 of genetics, this mutation insurance will prevent there from being any major changes to the organism's structure. So these are a couple of notes that are already on your sheet, but things that you want to remember. For this universal codon chart, there are a couple of things that you want to see. So you can see there, AUG, as is indicated on the chart, is our start codon. So whenever you see AUG, that's the start of a new sequence. That codes for methionine, which is an amino acid. Then there are 3 different stop codons, UGA, UAA, and UAG. These codons mean stop. So AUG means start reading, UGA, UAA, and UAG means stop. And we're going to do practice with this during class, so don't be too intimidated by this specific part. This actually seems more complicated than it is. And you can see the stop and start codons are indicated on this version of the universal codon chart. There's a lot of different versions of this chart, but they all say the same thing. So how are the codons matched to amino acids? This is where the tRNA that I keep bringing up actually comes in. So tRNA. tRNA comes in with amino acids attached. And the tRNA is what's going to actually drop off the needed amino acids, similar to how RNA polymerase actually drops off the RNA, the RNA bases as the DNA is being transcribed. The tRNA drops off the amino acids as the mRNA is being translated, okay? So the tRNA is sort of helping the mRNA actually create an amino acid sequence. And it sort of looks like this, right? So it is using an anticodon, which is the opposite of the mRNA sequence. So you can see there, AUG is UAC. It is still RNA, so there's no thymine, but it is the antithesis to the mRNA sequence. It is the opposite. So this is very similar to the process of DNA being transcribed into RNA, but using instead now, this is outside of the nucleus happening in the ribosomes, this tRNA or transfer RNA. This transfer RNA drops off these amino acids using the anticodon to link the chains of amino acids to create a protein. And you can see there, it is the anticodon. So there's the codon. GUA, for example, and the anticodon is CAU. And when we are using the universal codon chart, you have to like do this translation before you pair the amino acids, but we will do that practice in class. So just understand right now the terms. An anticodon is a block of 3 tRNA bases, and these are used to actually link the amino acids with the mRNA chain to produce the proteins. So this is why this part is different from transcription, and we call it translation, because we are converting between multiple different languages of genetic code to actually produce the protein. So we're using the mRNA as the template to take the tRNA anticodons, which will actually drop off the amino acids and link those amino acids in a chain to form a protein. This is the final step in protein synthesis until you get to a more complicated biology class, but this is the final step in protein synthesis that we will talk about here. I warned you it was complicated. So mRNA to protein, that is translation, which is the process that we just talked about. So the working instructions are the mRNA, and these are notes that you're copying on your paper. So it says the working instructions equals mRNA. The reader equals the ribosome, and the transporter is this transfer RNA. It might help. tRNA is the transporter. So what is actually happening? We have this strand of mRNA. It has left the nucleus. It makes its way to the ribosome. The ribosome reads the strand of mRNA, and then the tRNA comes in and drops off the amino acid sequences in order to produce a protein. Wow, fantastic. So from gene to protein, we have the DNA that's housed in the nucleus. The mRNA is what's able to actually leave the nucleus because that DNA is too important to leave the nucleus. We have to leave it in there safe and protected. Then that template strand of mRNA is the process from DNA to RNA is transcription. And then from the process of mRNA to protein, that is translation. First, the ribosome reads the mRNA, and then it is translated into a sequence of amino acids, which becomes a protein. And tRNA is the helper, which actually drops off the amino acids in order, very similar to how RNA polymerase is used during transcription, and also very similar to how DNA polymerase is used during DNA replication. And then those different amino acid sequences that become proteins are what make up our traits or physical characteristics. So the DNA in the nucleus is the blueprint that becomes the mRNA, transcription. Then the ribosome is the protein factory. That's actually going to be the site of protein synthesis. This is where translation occurs, taking the language of mRNA and turning it into the language of proteins, which is amino acids, with the help of tRNA that actually drops off the amino acids in sequence to create the correct proteins to build the structure of our traits and genes. And shwabam, we're producing a protein, baby. And those proteins become our traits, become the structure of our bodies. And that little penguin there is just real cute. Okay, so that was a lot. So I'm going to link an additional video about the process of transcription and translation, but it's kind of long, so you might just want to use this video to help you describe the process of protein synthesis using the words on your paper. So you need to use tRNA, mRNA, codon, amino acid, protein, DNA, gene, translation, transcription, nucleus, cytoplasm, and ribosome. I have included my description, but you want to write your own because that will help you understand this process. I'll show you just an example of what I do. So I made this as like my illuminated manuscript, if you will. This is how I make sense of things when I am trying to explain them. And I highly recommend, even if this isn't what you turn into me, you turn your handwritten notes into a short paragraph. try to use those words to write up your own description of this process because it is complicated. Like I said, this is the most complicated thing we're going to cover all year. So that's all I have for you today. Good luck writing up that description. Again, I have included my description. This is attached to the assignment on Google Classroom. So go back to Google Classroom and you'll see it in a PDF. Please do not use my description. Write up your own, even if it's shorter, but feel free to read mine for inspiration. Today's aim was, how do DNA and RNA assist in protein synthesis? And boy, we talked about the processes of transcription and translation. I hope you enjoyed that and learned something. Have a super fantastic day, and I'll see you guys all tomorrow on Zoom. Bye, guys.